Hi, everyone. Happy Friday, our last day before spring break, right? So I appreciate those of you who are tuning in and haven't yet mentally checked out for spring break. Okay, let's talk more about the special surfaces inside of our body that are known as the mucosal layers, right? And how our immune system evolved to sort of treat these specialized surfaces differently than the physical barriers that are like our outer skin. Right? So we said that so far in the lecture, when we've talked about understanding the basics of innate and adaptive immunity, for convenience sake, we've been referring to when things like microbes get in through cuts in our skin. And that's helped us to sort of understand some of the basics about how proteins and certain types of cells work together to mount an innate immune defense, and how that can lead eventually to the adaptive system. We also said that as we talked more about the adaptive system, we realized many of the examples that we would use are things like viruses, right? Novel viruses. And of course, those, a very common point of entry into the body is through your respiratory tract or through your GI tract. And that's what led us then into closer examination of the types of physical barriers that line those surfaces. Right? And we said that the mucosal layers, the layers of the different tracts in our body, not only our respiratory tract and digestive tract, but also things like our urogenital tract, basically any type of surface that technically is forming a boundary between inside of our tissues and outside of the tissues that are not an obvious external surface that you sort of can see in the mirror, right, involves most likely some sort of mucosal layer. And they have a thin layer of mucus protecting them. And importantly, we said those layers are very thin, very delicate, because they have to be in order for them to carry out functions like gas exchange or nutrient exchange, etc. So last time we began to appreciate that those surfaces oftentimes have secondary lymphoid tissues immediately surrounding them, right, or immediately under them within our body. So this was the slide that we left off with last time, and it was to highlight the differences in how systemic immune responses dealing with something like a cut in your skin and bacteria getting through that way, right, compared to infections involving your mucosal layers. For example, this cartoon shows maybe a region of your small intestines. Right? We said, importantly, right, structurally, even though the cartoon doesn't really show it very well, the skin is tough and impenetrable. There are several layers of keratinocytes. I know they only show one up here, but right, most of this pink stuff in here would also be skin cells. And then they don't show that you have this tough multi-layers of sort of dried keratin proteins on top of it, right, compared to something like a mucosal layer typically found like in our small intestines that would quite literally be like one cell layer thick, very delicate. And the blue space up here is not like the outside air that surrounds you right now, but this is the space inside of your intestines, the lumen, right, where all kinds of food and other things are passing by. In both situations, you would expect that there'd be some population of bacteria present, right? But in the outer skin, right, these bacteria are just kind of spread out, and it's just kind of a function of what's around you in the environment. If you do get a cut and the bacteria comes in, we learned that one important mechanism of your innate immune system is that if macrophages, complement proteins, cannot within just like a couple hours take care of the problem, right, they are busy sending out cytokine messages, and that will cause your induced innate reaction. So things like more complement proteins will leak out of your capillaries. The capillary walls become leaky to make it easier for more proteins, even things like Igs that might be in your blood so that they have an easier chance to get into the tissues, and importantly, entire cells. Things like neutrophils or more macrophages can squeeze out. And we said that inflammation in this scenario makes a lot of sense, right? Because by the surrounding area becoming inflamed, the local temperature goes up a little bit. So maybe just a couple degrees is enough to cause the bacteria to slow down in the replication. Also, it gets very firm and a little bit swollen to the touch. That's because not only is more fluid draining out of your leaky capillaries, right, but the fluid is having a slower time draining away from the region, right? So that way you're discouraging things like bacteria from spreading. So because of this inflammation, there is going to be increased tissue damage at this area above and beyond what is just caused by the bacteria itself. So the inflammation and the side effects of inflammation, the tenderness, the swelling, right, a little bit of physical tissue damage is actually part of your immune system and its way of trying to keep that bacteria there, slow it down, and neutralize it. So as a result, you will have to, have some healing involved, right? And it might involve a scab and a scar, etc. So hopefully that makes sense to you in the context of everything we learned about how our sort of systemic immune system works. But when we talk about the mucosal linings in our gut or in our respiratory tract, right, it's a very different set of considerations. Number one, that layer is very delicate to start off with, so not nearly as physically imposing as our outer skin. Right? Secondly, because of the environment of things like inside of your GI tract and your respiratory tract being moist, right? And there's lots of nutrients and things like that, especially in your GI tract, you would expect to have lots more bacteria in your gut than you actually do on the surfaces of your skin. Right? And so if you have things like bacteria that are able to get through the mucus layer, and I remind you, right, these little bow tie shaped immunoglobulins are dimeric IgAs, right? They're IgAs that have dimerized to form this little bow tie. And that is the majority of the antibodies that are in your mucus are the IgAs. Right? The majority of antibodies that are floating around inside your blood are IgGs. Right? And so some of these bacteria, maybe they do manage to get through the mucus, right? Maybe because for some reason things are getting out of balance here, and there's way more of this bacteria sort of blooming, if you want to say, in your gut than normal. And some of them make it through, right? So the critical thing here is that, yes, there may be some inflammation of the tissue as a result of the bacteria itself damaging the tissue, right? But your immune system does not want to increase the inflammation, right? Because the last thing you want is this already delicate mucosal membrane to be compromised even more, right? By swelling or leaky blood vessels, things like that, right? Because then all of that bacteria, that normal commensal bacteria that's in your gut, right, suddenly would just come flooding in to your tissues, right? And ironically, that would be even worse potentially than the initial infection of whatever bacteria was getting in by itself. So as a result, things like macrophages, so as a result, right, this underlying tissue that surrounds our mucosal layers is filled with immune cells already, right? And so that's why we said that this is a specialized kind of lymphoid tissue. We call it the mucosal associated lymphoid tissue, or MALT. But again, because the GI tract presents its own set of additional challenges, right? Like having an abundance of bacteria sort of expected to be in there in the first place, right? The MALT of our GI tract, we kind of give a special name. We call it the gut-associated lymphoid tissue, or GALT, right? So GALT is just a specialized type of MALT, and MALT itself is just a specialized type of lymphoid tissue. You can think about it this way. On the top slide, right? These immune effector cells have arrived at the area, or they were already there, to kind of help fight this localized systemic infection, right? Where are the lymph nodes in this? The lymph nodes would be somewhere off of this picture, maybe some distance away, right? Because remember, if this infection gets really bad at this site, and even the induced recruitment of things like neutrophils, whatever, can't neutralize or beat the infection after several days, right? Eventually, whatever lymph node nearby, right, would be receiving stuff that's draining in there through the lymphatic vessels that are draped over things like this capillary, right? So here's a capillary here. So draped over this would be one of those big green, the cartoons show them as green lymph vessels, right? So over time, somewhere off the picture here, there'd be a lymph node where lymphocytes like B cells and T cells are hanging out, right? And that's when they have a chance to be selected and potentially activate. But in the lower picture here, right, your malt itself, or the galt itself, is like the environment of a lymph node, right? It just spread out over everything, right? So you have a whole abundance of lymphocytes already there and ready to work. And so effector cells like the macrophages here are different than the macrophages, or I should say they behave differently than the macrophages that are in the top part of the cartoon, because instead of inducing inflammation like you see up here in the top part, right, these malt-associated macrophages, when they ingest or destroy bacteria, they do not send out inflammatory interleukin cytokine messages, right? It's almost like your body is confident that just the immune cells that are already in abundance in that surrounding malt tissue is enough to neutralize the pathogen. And so no need to cause inflammation that might damage and make things worse. Notice that there is a draining lymph node shown on the cartoon here. I think because it's a reminder, right, that even though this whole yellow region is functioning sort of like a lymph node, right, you still want the ability of things like dendritic cells to actually take stuff and go to a systemic lymph node so that you have additional opportunities for things like B cells to be selected, T cells to be selected, in case you really need to eventually wake up an adaptive immune response. So we said that the lymph nodes that are receiving the stuff coming from your gut, the galt tissue surrounding your gut, we tend to call those mesenteric lymph nodes, to separate them out in our minds from the other lymph nodes that are part of our systemic immunity, the lymph nodes that are part of your throat, underneath your jaw and your throat, or under your armpits, right, or in the pelvic region. So what is mesentery? So mesentery, I mentioned this last time, but I think it's more useful for you to see a picture. Those of you who have studied human anatomy, well, even those of you who have not, I think you're aware, right, that the intestines, like the small intestines, large intestines, they're just not like piled up inside of your gut, and they just kind of like sit on the bottom of your gut, right? I mean, that doesn't really make sense. I think you're all aware that they're somehow suspended in a way that makes it or facilitates the passage of food through them. And there's certain parts that actually go against gravity, right? Stuff has to come up and down, like in the large intestines, right? So therefore, they are actually held in place by this network of membranes that almost provide sort of like a scaffolding to keep everything together. And so this is the mesentery, this big set of folded membranes. So this yellow space in here is the mesentery. Behind it is actually a lot of your small intestine. And you can see at a certain point, the small intestine actually comes through the mesentery, it's coiled up around here before going to the colon. And then the colon, there's an ascending and transverse and descending colon region. They are held in these positions because of connections with these membranes. What's kind of interesting is, I think, what is it, maybe 5 years ago, something like that, I saw online there was this big news article about how scientists had discovered a new organ in the human body, right? And I was like, well, that's crazy, right? Because you would think by now we know everything that's in the body, right? But it turns out what they were saying was a group of European researchers, after doing a lot of work on this and really looking at the layout of our gut, concluded that the mesentery, instead of being a whole series of separate sort of folded membrane connections, actually they considered it one continuous sort of like membrane organ that is wrapped around everything, right? So functionally, they didn't treat it any differently than, let's say, your liver or intestines or heart or lungs, right? Because I think to us, it's obvious your lungs are an organ or a pair of organs. Your liver is a separate organ. Your heart is an organ, right? So now they consider this mesentery as being another organ in the body. I mean, I was kind of disappointed because I thought they were going to say they like found something that was like hiding in the brain, right? And then I thought, oh, good, I can use that as an excuse for why I am the way I am, right? No, so it turns out it's just the mesentery, right? But what it does highlight is how important the mesentery is, right? That it actually functions as a separate organ. Within this yellow region on the cartoon, you can see the green things are lymph vessels. And so what this shows you is how extensive the lymphoid drainage system is around your GI tract, right, around the intestines. In other words, your body is constantly monitoring what's going on inside of your intestines. And I hope that makes sense to you, right? Because after all, that's an example of us every day on purpose putting stuff right into our GI tract. And there's an undeniable opportunity for pathogens to gain entry, hitching a ride on things like food, right? So it should make sense that our body really, really monitors what's going on inside of our intestines very carefully. And so there's these special network of lymph vessels that drain into special lymph nodes. And these would be the ones that we call the mesenteric lymph nodes, right? Because they receive information, samples of stuff like antigens coming from your gut. Okay. Let's look at a scenario here where, remember, we have these specialized little regions, little spots in our small intestine, and only in our small intestine, called Peyer's patches. And these are the, we have like 100 or 120 or so of these little domes. And keep in mind, I mean, this is still microscopic, right? This is just like a little dome of cells with a little sunroof. These are the M cells that on purpose allow bits and pieces of stuff from the lumen of your small intestines to come into the tissues, right? And literally, this is on purpose. You are encouraging infection to happen, right? But it's okay because these Peyer's patches are where you have tons of immune cells ready to receive these samples, right? And so you can see here's a high endothelial venule sort of entering the picture, and you have different lymphocytes exiting here, and they can check out what these dendritic cells are showing them, the little red dot. They will then drain right through those lymph vessels in the mesentery, right, into these mesenteric lymph nodes. And in these mesenteric lymph nodes, then you have the opportunity, right, for lymphocytes to, like naive lymphocytes to activate, just like we talked about before. So the activities inside of a mesenteric lymph node are not that much different than the activities we've learned about in other lymph nodes, like our systemic lymph nodes. Importantly, though, when those lymphocytes activate, let's say the blue ones are, I don't know, I guess these are T cells, the yellow ones are naive B cells, right? They drain out of the lymph node, they go through your big lymphatic system, right? Even though the cartoon shows that they only go a few inches, that's not true, right? They go all the way through your lymphatic system now, up back to the level of your shoulders, where they get reintroduced into your blood, right? And then they get pumped by the heart through your whole system, your circulatory system. Right? So some of these activated lymphocytes will make their way back through your blood just by chance, right? Back to, let's say, the small intestines, right? So that's why they ended up kind of next door to where they started off. And you can see those T cells and B cells will squeeze out of the blood supply, out of the capillaries, right, through this process of diapedesis. That's when they squeeze out between the cells of the wall of the capillary and get into the tissue, right? At that point, the B cells, right, they go ahead and differentiate and turn into plasma cells, right? So they can go ahead and start releasing antibody, right? In response to, let's pretend that this red dot up here was something that, you previously had developed like an immune response to, right? So you have immune memory about this, and that's why these immune cells, right, are sort of triggering this memory. And so this plasma cell then starts making way more IgA, so that the IgA can get into the mucus up here and really start dealing with, right, whatever this pathogen is, virus, bacteria, whatever. These T cells, right, you see they come out, right, and they're active. Notice how many of them will go ahead and actually squeeze their way and insert themselves, plant themselves in between the epithelial cells of your mucosal layer, right? As though they are standing guard, right? And they're on alert, they're standing guard, right, to receive any evidence of cells surrounding them being infected, right? I want you to also keep this in mind. Even though the cartoon suggests that this particular little group of cells made their way back to the small intestines, right, so that they could actually start protecting the small intestine region over here, the reality is, even if stuff was detected coming through the Peyer's patch and activating your immune memory or selecting for new immune cells, right, those immune cells, as they go through your blood, they're not just going to head back to your small intestines. They will exit your blood throughout your mucosal layers. So even though this was your small intestines, your gut, you will have these B cells and these T cells also exiting the blood to take up guarding your respiratory tract lining, right? And you say, why is that? Because evolutionarily, right, if this is a pathogen that you ate through your mouth, right, it's quite possible that this is also something that you might be breathing in, right? And therefore, the linings of your respiratory tract also want to go on alert, right? So these B cells and T cells will start to protect all mucosal surfaces throughout your body in response to just something like this being detected in your gut. It's like better safe than sorry, right? So this is a nice slide because it kind of summarizes the types of participants in the mucosal immune response. So again, this is your gut lumen. So all the white space on the right part here is the space inside of your intestine. So you have a very thin, delicate layer of epithelium. Okay. Right beneath it, you have the specialized mucosa associated lymphoid tissue, the MALT. In this case, we call it the GALT because it's the gut that we're looking at as an example, right? And it's filled with things like macrophages, special macrophages, right? That if they actually eat pathogen, they won't send out inflammatory cytokine messages. There will be dendritic cells. These are more or less just like the normal dendritic cells elsewhere in your body. They want to scrounge around and find evidence of stuff to show the other immune cells. We said that the dendritic cells are very funny here because they're a bit like over anxious. They're really eager, right? They will actually reach between the epithelium just to hopefully sample stuff on the outside, right? You also have what was I going to say? Other granulocytes like mast cells. Right? Remember, we learned that certain granulocytes, mast cells, eosinophils, basophils, their job is normally as kind of like an innate effector cell. How do they work? They work by storing large amounts of all kinds of nasty sort of nasty kind of substances that encourage strong tissue reactions to happen. Right? So histamines, for example, right? Histamines that cause you to sneeze, cough, whatever, right? Have an itchy throat to try to get you on a very sort of macro scale to have some sort of reflex like cough or sneeze or whatever to try to get stuff out of you, right? So those would be hanging out there as well. And remember, we learned that even though normally something like a mast cell is innate, right? It doesn't really care what it's sort of directing its attention at. It just, when it's triggered, it'll go ahead and just release all this stuff in a big cloud, right? We learned how mast cells and other granulocytes have certain receptors on their surface, Fc epsilon receptors, that can hang on to the stems of IgEs. Right? And therefore, being coated by IgEs, it's like they're being suited up, right? They're just sort of normal, everyday mast cells, right? But once they're suited up with IgEs, then they have additional powers because the IgEs will actually get them to specifically react against antigen targets that the IgEs are designed against, namely things like large pathogens, right? Like a mast cell isn't going to really care if there's virus particles, right? A mast cell might not even care if there's bacteria, because the bacteria, remember, scale, right? The bacteria on this cartoon would be teeny tiny compared to the mast cell. A bacteria would be even smaller than one of the purple vesicles inside of the mast cell, right? No, the mast cell is there just in case, right? You ate some nasty like worm or something like that in your food, right? Or you know, I don't know, the eggs of the worm or something really gross that are now kind of like causing an infection, a parasitic infection in your gut, right? That's what the mast cell is there for. So that if all of a sudden you have an infection by a big worm coming into this cartoon here, right? This mast cell and its other buddies who are coated up with or suited up with IgEs, right? They go ahead and stick to the worm, and then they all release their awful, you know, molecular, you know, irritating kind of things in an attempt to really, really harm that worm, right? Okay. In addition to that, we have plasma cells, right, that are releasing IgA. And then we also have things like CD4 helper cells and CD8 cytotoxic cells. And I should point out, I did not elaborate on this when I first showed you to this. It's the CD8 cytotoxic cells that take up residency in between the epithelial cells, right? Because they're the ones that want to really stand guard. They're like little sentries at sentry posts along the way, protecting this wall, right? So if any of these cells that make up the epithelium start showing signs of infection and present the antigen that the CD8 T cells have the T cell receptor for, right? They can go ahead and quickly start killing off these epithelial cells to discourage the spread of, let's say, a virus. Right? All right. So this is a nice summary table to help you appreciate why the mucosal immune system functions differently, right? Or how it functions differently compared to our systemic immune system. And there are some things you can think about. You can think about the way it's kind of set up anatomically. You can think about the way effector mechanisms like cells and things like that actually deal with an infection. Right? And you can think about the environment itself, the tissue environment, right? How that is different and how the environment, the handling of the environment is different because you're dealing with mucosal tissue. We can talk through this pretty quickly. So certain anatomical features, right? The mucosal layers of our body have intimate interactions between the epithelium that actually represents the thin physical boundary between inside of your tissue and outside of your tissue, right? Very intimate contact between that and specialized lymphoid tissue right underneath. So that's different than our systemic system that really, you know, like under your skin, in order for stuff under your skin to really get to any sort of lymph node, right? It has to drain and actually make its way, you know, to some nearby lymph node. Like if the cut is in your arm, maybe the lymph nodes in your armpit are what's going to ultimately be the place where B cells and T cells are activated, right? So that's different. There are discrete compartments of diffuse spread out lymphoid tissue, as well as little organized structures like the Peyer's patches, right? Or things like tonsils. We talked about that. That's another specialized anatomical feature as part of your mucosal immunity. Right? And thirdly, the way the immune tissues sample stuff, right, involve special cells. These are the M cells that allow antigen to actually on purpose enter tissues. You know, I described it before as it's kind of weird. It's almost like on purpose you're allowing some infection to happen, but it's okay because you're ready and expecting it, right? So that you can quickly neutralize the pathogen, right? And yet benefit from having a sneak peek at what this pathogen was so that your mucosal immunity can kind of be alerted. Uh-oh, right? Here comes salmonella again, right? And also, I was going to say that, what was I going to say? I was going to say something, and then I thought about salmonella, and I got distracted. Yeah, and so importantly, right, that's a bit different than the way our systemic immune system works. Then the next part, effector mechanisms. In your mucosal tissues, you will have lots of activated T cells, right, ready to go, even if there's no infection going on, right? And these T cells, they're hanging out there. Some of them are kind of like in between the epithelial cells, just standing guard, right? So that's very different than how, like, what you would expect underneath your outer skin, right? And you'd also have plasma cells hanging out, right? Plasma cells that are hanging out there so that right away, right, if a pathogen comes in, antigen gets presented, and this is the antigen that these plasma cells, were selected to recognize, then right away you can have big release of things like IgA into the mucus, right, to sort of start preventing more of the pathogen to get through the mucus and into the infection. And then the environment itself. Most importantly, it's that issue of inflammation, right? In our mucosal layers, we discourage inflammation as part of the immune response. I said before, there's always going to be some inflammation just because there's going to be tissue that's damaged, right, as a result of the pathogen breaching and making its way in, right? The bacteria starting to multiply, sending out all kinds of really obnoxious kind of like enzymes and proteins to try to remodel your tissues to make it happier living there, right? Or viruses infecting cells, and the cells then lysing and releasing, more virus particles, and at the same time having an interferon response, right? Alerting your immune system, oh no, we have a viral response, right? Both of those scenarios are inflammation as a result of tissues being damaged, right? But what we're talking about here is the immune response itself does not want to add to that inflammation, right? Because that might make things worse if you're dealing with the environment of your mucosal layer, right? And so many cells like macrophages, and dendritic cells, they behave slightly differently in response to picking up pathogens in the mucosal layer. Right. So here's a question. A couple of you have asked me about, every single day we eat all kinds of stuff, right? And clearly there's all kinds of stuff that is foreign to what we naturally produce inside of our bodies, right? Many of these substances could actually be very suitable three-dimensional shaped molecules that would serve as antigens that our immune system might have a very easy time kind of seeing as an antigen to bind to and then react, right? So why is it that you can actually eat all kinds of stuff and yet not always have this tremendous mucosal immune response, right? Clearly, that would not be compatible to our daily lives, right? If after every meal that we had, right, we have to sit back and kind of suffer through some sort of strong immune reaction as our immune system somehow sorts through like what's really going on in there, right? So oral tolerance, okay, this word tolerance we've used before, right? Tolerance means what? Your immune system knows to kind of accept something as being okay. We talked about tolerance in terms of B cells and T cells that are developing, having to undergo tolerance training, right? So that B cells and T cells that, because of the shape of their IGs or T cell receptor, happen to bind to self antigens, right? They need to be prevented from going to work because otherwise you'd react against yourself, right? So that's self-tolerance. So oral tolerance, same idea. Your immune system can't freak out all the time just because you're eating something that contains antigens that did not come from you, right? So how does it know the difference? This is an interesting kind of statistics here. A typical fast food meal contains about 300,000 living bacteria and yeast and mold particles in it. All right, I'm not even talking about some restaurant that's not clean. It's just, typically anytime you have prepared food that's sitting around, there's going to be stuff that starts, falls on it and starts kind of growing, right? 300,000 living bacteria, yeast, and mold every time you have a fast food meal, right? I'm not discouraging you from having a Big Mac meal, right? Because that's actually, I'd be a hypocrite if I was, because I like that very much. But the whole point is, it's okay, because that's what we evolved to kind of expect. You know, no, that's not a good example, because we did not evolve to expect a Big Mac, right? But we evolved to expect that every time we're eating stuff, there's going to be pathogens hitching a ride. So everything in your intestines, right, whether it's the food you intended to eat or the pathogens that hitched a ride on that food, everything gets broken down into little bits and pieces of molecules, right? Some of it's nutrients, some of it are actually pathogenic antigens, bits and pieces of these guys up here. And so they all get sampled by the M cells of your Peyer's patch, right? And your immune system has a crack at trying to, bind it and see what's going on. So why don't we have an immune reaction every time? Again, this has to do with one of the fundamental differences between systemic versus mucosal immunity, and that is the role of inflammation. Antigens alone won't activate a GALT response. That's because, as part of our mucosal immunity, right, our mucosal immune system works very hard to suppress inflammation. And hopefully, you've learned by now in this semester that inflammation is a big part of your immune system kind of waking up and taking something seriously. Right? So let's imagine in your gut, you're eating the Big Mac, right? And bits and pieces of Big Mac antigen, right, are broken down in your small intestines and absorbed and sampled through the little M cells and the Peyer's patches. And then you have dendritic cells that pick up on it, right? So they're showing the little red dot here, except in this case, let's pretend the little red dot is some sort of antigen that comes from the burger. Right? So let's say you happen to have a T cell that's hanging out there, right? And this T cell happens to have T cell receptor that is the right shape to bind an antigen that's being presented as a result of you eating this Big Mac. And so this is like a naive T cell, right? It's never bound something like this before, right? But suddenly, oh boy, right, its T cell receptor picks up on this that's being presented by the MHC of this dendritic cell. But what? Right? Because there's no inflammation going on, right? The cytokine soup in this tissue, right, is not conducive to the T cell activating, right? Because this is just a normal meal. So the dendritic cell, right, shows this, and this T cell comes along and says, hey, that's what I recognize, right? But the dendritic cell says, nah, it's okay. It's just lunch. It's fine, right? And as a result, this T cell then, right, as part of this oral tolerance training, right, is eliminated, right? It becomes anergic. It's sad, right? It goes off, right? However, let's say you really have an infection going on, right? Let's say, for example, right, that on the Big Mac, there actually was some like bad E. coli because the patty was undercooked or something like that, right? And then a few hours later, or actually, I would say maybe a couple of days later, enough of this googly-eyed virus particle, right, or bacteria, right, I said E. coli, begins to multiply, right? Therefore, in your malt, you're picking up things like, the little red dots are actually coming from, the bacteria here, right? And so if this T cell recognizes the red dot, I don't know why the dot became yellow here, but, it's the red dot, the peptide that's coming from the pathogen, right? The T cell says, hey, that's the peptide that I recognize, right? And what does the dendritic cell tell it? There is an infection going on, right? And this is one of the many peptides that we're seeing as part of this infection, right? So therefore the T cell says, all right, right? So it activates and initiates your adaptive immune response, okay? So this raises an interesting scenario because where's the Big Mac in this picture? Well, it's not in the picture, but the whole point is, what if antigens from the Big Mac are mixed in here as well? Right? And if those antigens are also being presented on here, and that other sad T cell, right, came along, and before it went anergic and it was told, don't worry about it, if it went ahead and bound to this dendritic cell 2 in response to the peptides that come from the Big Mac, it would activate as well, right? Because the dendritic cell is saying, hey, there's an infection going on. All these little bits and pieces of peptide, we don't know what they are, but this is what we are seeing right now, right, as part of this infection. So any of this stuff, right, could be important for you T cells to pay attention to, right? And what could that lead to? That could lead to a form of food allergy, right? Because maybe you would start selecting for immune memory cells that know and recognize certain peptides that come from the food part of this and not the actual pathogen part. So I want to tell you a story. Okay, so it's kind of a short lecture today. So I figured I'd sort of end with this story here. Oh, right. So I wanted to point out, the topic of food allergies. As human beings, especially in modern society, we eat all kinds of things every day, right? We evolved to eat a pretty limited range of different foods in our normal lives, right? I mean, we're omnivores, so we eat things like plant material. We are capable, or part of our normal omnivorous diet is to catch things like small animals, right? Fish, other mammals, things like that. But if you think about it, right, we evolved not to wander too far from our, little village or whatever, right? And so the typical diet from day to day to day probably wouldn't vary that much, right? I mean, maybe one day some of your friends wandered, a few extra miles in one direction, and they come back and they said, we found these berries or something like that, right? And then it's like a huge treat, right? But then by and large, every day you're eating the same stuff, right? In our modern society, think about how crazy it is, right? That in the morning you could wake up and be like, okay, I want to eat like an omelet, or something like that. Maybe at lunch you go to McDonald's, right? And for dinner you're eating, pizza, burrito, Italian food, Chinese food, Thai food, Vietnamese food, right? Local Hawaiian food, right? All that stuff, right? Such a variety easily at our disposal. So all kinds of antigens get mixed up every single day and vary day to day. Right? I think some of you know there are certain types of foods that you personally say, yeah, I don't do Thai food very well because it doesn't agree with me, right? Or I don't like, that kind of food because last time I ate it, it made me sick, right? Could it be, right, that it's actually not because there's something like a pathogen in that food? So if you eat it again, you actually have an immune response against the pathogen? Could it be that there's actually certain types of peptides or certain types of antigens, just part of the food itself, right, that you've improperly developed immune memory against? Right? Most of those types of antigens will be small peptides, right, just like in this scenario here. It turns out that a very, very common and abundant source of widely varying small peptides that are highly antigenic come from different spices. So different spices that we put on different types of food, Italian spices, Asian spices, right, whatever, right, all have different sets of peptides on them, and all of them are potentially antigenic. So that's why you might say, I don't do Italian food well, because when I do Italian food, I get a stomach ache, right? It might actually be at some point along the way, it's because you developed immune memory against certain types of peptides that are present in the spices of Italian food. Right? Okay. So that would be a case of what? Almost like mistaken identity, right? Because obviously there's no reason for your immune system to really be worried about detecting Italian food, right? But think about what we just talked about, right? The example of the Big Mac, right? Maybe somewhere along the way, one time, a meal that you ate of a particular food did have some sort of pathogen in it. And in the course of your immune system learning how to recognize signals from the pathogen, right, it could not really distinguish those from those brand new different types of antigens that it was also seeing in things like the different spices of that particular dish. Right? All right. Quick story at the end having to do with this. Um, so this Starbucks is actually the Starbucks that's located in the Longwood Medical Center in Boston at the Harvard Medical School. And so when I was doing my postdoc there, my lab, or the lab that I belonged to, was in the building right next door. And so all the time, grad students, postdocs, we'd be hanging out at this particular Starbucks. We're supposed to be hanging out in the lab doing work, but whatever, it's way more interesting hanging out at the Starbucks. And so I remember one day I was in the Starbucks, and I usually back then, I used to really love getting those caramel macchiatos, right? And one day I thought for some reason, okay, I'll change it up a bit, right? Instead of the regular milk in there, I'll ask for the soy milk, right? So I'll get the soy caramel macchiato. And so the barista makes this, and I'm drinking it. And as I'm kind of sitting there, just within a few minutes, I start having this funny reaction, right? I feel like my throat's getting itchy. It's beginning to tighten up. And I started feeling like I started wheezing, right? So I'm like, oh, this isn't good. I don't know what this is, but I don't know what's up with this. So I go back up to the building and go into the lab, and I go to my PI's office, right? My boss, and my boss, he's this really world-renowned prostate cancer researcher. So he's most of the time at his desk with his phone, whatever, talking to other worldwide famous researchers, right? And so I go in there, and he looks up, he's on the phone, right? And I say, Steve, I'm like wheezing, right? I'm like, Steve, I think I can't breathe anymore, right? I think I'm gonna pass out. And so he kind of looks at me and he just says, sit down, right? And so I'm sitting there in his office, and by now, right, I'm really having trouble breathing, and my eyelids are all puffing up, right? So that's strong, and I'm getting kind of embarrassed, right? So I'm kind of laughing, wheezing because I'm all embarrassed. And I'm like, I really don't think I can breathe. And he says, so he's on the phone, right? So he covers the phone. He's like, don't worry. I mean, look, we're in a hospital, right? So if you actually fall down, I'll just get one of the other grad students to wheel you down the hall, right? And I'm like, oh, okay. So this goes on for about 20 minutes, right? And then after a while, I felt better. And so I said, all right, I'm going to go now, right? And I think Steve's like, okay, good, because now, I can get back to work, right? So I was really kind of scared, right, of that strong reaction, because I usually don't have strong reactions like that to food, right? And so I was really wondering, what is it, right, that in that soy caramel macchiato that could trigger such a fast immune response? So, right, what is the nature of that response, right? It has to do with mast cells lining my GI tract that very quickly detected some sort of antigenic signal that was in that caramel macchiato, the soy caramel macchiato, so that right underneath in the lymphoid tissues surrounding my GI tract, all those mast cells, right, because they have IgE on them, IgE that recognize something in there, right, that previously it means my body must have learned was a threat, right? And so all those mast cells started exploding, releasing all these histamines and things, causing my GI tract, right, to constrict. And what? All throughout the other mucosal layers, for example, surrounding my respiratory tract, those mast cells were also releasing histamines, right? So it was a real overboard immune response, but I was mystified what it could be. Obviously I thought it's probably the soy, right? Because that's the only thing that's different. But then I thought about that and no, that doesn't really make sense. I can't be allergic to soy because, I'm Chinese, right? So being allergic to soy is some sort of Darwinian, evolutionary dead end or something like that, right? So anyhow, when we get back from spring break, I will tell you the rest of this story. Right, so it's like a little cliffhanger to leave you with for spring break. And I think you will find it very interesting and enlightening, and hopefully it'll help you kind of understand how the things that we are talking about really relate to your own experiences every single day. Okay. All right. So have a great spring break. And I will see you when we resume classes. All right. Bye-bye.